What I'd like to do this morning is to talk to you about a project we've been working on for the last couple of years, having to do with attempting to identify new therapies for a particular autoimmune disease, SLE, and we've gotten involved in doing this with a drug repositioning effort. And what I'd like to do is to review with you a little bit about some concepts of drug repositioning and then get more into the approaches that we've done, which we think have the capability of generating new therapies for this disease, which many of you know has had only one new approved therapy in the last half century. So drug repositioning, which in the literature you can see called drug repurposing, drug rescue, drug reprofiling, retasking, resurrection, a variety of other things, is something that's well known in the pharmaceutical industry. When a company has a product and the product doesn't meet its primary endpoints in its initial clinical development, or for some reason some person with an expensive suit decides not to invest in the program, or the patent runs out, or the drug is still in development, many times an effort to rethink how that drug might be used is made in industry. And this has generated a very, very extensive field of study of exactly how to go about rescuing drugs or repositioning drugs that may have not done well with their initial indication. Now, in diseases like lupus, the condition may actually never have been seriously considered during drug development. And that's true of many, many drugs. There are some companies that think seriously about lupus, but as we go forward in this field with so many failed trials and such an uncertain pathway to approval, many companies really haven't considered lupus at all as a condition. So what you might consider external repositioning, those from the academic community looking at the characteristics of drugs and trying to identify ones that could work in this condition, and perhaps working with their partners in pharma and biotech to reposition them for this disease. Now, within industry, the reasons to consider drug repositioning is what's been called in the literature the innovation gap or deficit. And this is really the widening gap between productivity and the R&D budget. As you all know, the number of drugs that are approved each year is diminished, although last year there was a bit of a bump, and the R&D budgets are becoming enormous. And this is, again, stimulated pharma to think very intensively about how to do the best they can with each asset that they develop. So the rising developmental cost, and as I mentioned, the reasons to consider drug repositioning from the academic position is that frequently drugs were never really considered during the development process. Just to point out this innovation gap, these are data, published data, going out through 2005, and it really hasn't changed that much since then. And the blue line is the cost of R&D development, and as you all know, it costs somewhere around $2.5 billion to develop a drug. And the tan bars are the number of new chemical entities that have been approved over the years. As I said, in the last year, the number has increased, I think, to about 40, but still the R&D costs have gone up during that period of time. So we have this tremendous deficit that has really caused pharma to rethink every asset they have. I think it was just published that in pharma, for every dollar that's invested, one can anticipate a 70-cent return on investment. So that's not a kind of equation that allows you to be casual about drug development and causes pharma to rethink carefully every asset that they have in the pipeline. Now, the central concept of drug repositioning is that you can wind up with new drugs from new knowledge or new analysis or new thinking about knowledge, but not necessarily from new chemistry. So when it works, which it does occasionally, it's a very, very efficient way to move forward. The advantages is that if you can find a candidate to reposition, it frequently has passed a number of significant preclinical tests, including toxicology, and if it's been in clinical or man, the safety is known, so the risk for failure for adverse events is mitigated, and many of these early developmental studies are completed. The challenge, of course, is that you're frequently downstream in the development process, and sometimes you're looking at patent life as a limitation in development, although there can be extensions of patents based upon new information. So just to review with you, and again, this is a paper published in Nature Reviews of Drug Discovery, but the typical pathway of drug development is long, estimated to be anywhere between 10 and 17 years. It's now estimated to cost about $2.5 billion, and less than 10% of drugs make it through this entire pathway. When you're thinking about drug repositioning, you've already passed all these steps, and basically you're starting here with whatever clinical development is necessary and regulatory development. The time can be cut down to as low as 2 or 3 years, and the risk is considerably less because many drugs fail because of their adverse event profile. So there are many examples of successful drug repositioning. I guess the most famous is Viagra, which was initially developed as an antihypertensive. Of course, minoxidil, which you can purchase to promote hair growth, and a number of other examples. In some situations, companies even invented diseases to reposition drugs into. But nonetheless, this is a it's been a rich source of drugs. Rheumatology, of course, where we've been repositioning since the very beginning. And for example, in lupus, nearly all the therapies except glucocorticoids really were repositioned from some other entity. Of course, we know of methotrexate that started out as a cancer drug, used now in many diseases. Rituximab also started out as a lymphoma drug and useful in a number of rheumatologic conditions. The antimalarials in RA and lupus, cyclophosphamide, mycophenolate, and thalidomide, along with others. So as rheumatologists, we're very familiar with this concept of drug repositioning. Now, drug repositioning years ago, and for many of the conditions that I mentioned in rheumatology, resulted from a bright idea or from clinical serendipity. Viagra developed because of an adverse event seen in clinical trials. Many situations occurred where clinical benefit was noted in individuals in clinical trials who happened to have a second disease. But now there's a much more formalized approach involving efforts to search large databases in order to understand and de-risk the whole process of drug repositioning. So the idea nowadays is that if we're smart enough and can analyze enough data, we can find drugs that were meant for one condition and now might be useful for another. So how do we go about doing this? And there are lots of different ways that one can do it. And I won't go through all of these, but there are algorithms that have been developed in order to approach either gene expressions or genome association studies, various pathway analyses. Some have developed algorithms to look at adverse events on the assumption that the adverse events reflect the action of the drug, and that by comparing adverse events, one can find new therapies. And then there's always literature mining. So I'd like to talk about two things that we've been involved in lately. The first involves a literature mining search and the second really involves a search of gene expression in order to find new drugs. But in the literature, you can find all of these other pathways that are involved. And the goal, of course, is to find new and better drugs, not more of the same. So we're really looking to find things that might be useful in this particular disease, lupus. So let me go through this literature mining approach first, because we've had some success with that, and I'd like to show you how we've done it and how we've progressed with this. And this began really as an effort funded by the two big voluntary organizations that are involved in lupus research, the Lupus Research Institute and the Alliance for Lupus Research. And they had been funding research for more than a decade in basic science, and were getting feedback from their donors that how come there hadn't been a faster translation of these basic science concepts into new therapies. And in thinking about how these voluntary organizations, which are not huge, might be able to enhance the rate of development of new therapies, a concept was that they could promote repositioning of FDA-approved drugs on the assumption that very few of these drugs had actually been considered for lupus in development. So I should point out that this was called the Lupus Treatment List. LRxL and STAT was the second phase, which we're moving into now, which is the Systemic Lupus Treatment Acceleration trials, and I'll talk about that a little bit. So 2 years ago or so this began, and the idea was to identify drug candidates and treatments that might be useful for lupus. And 4 approaches were taken. The first was to look through the entire database of drugs approved for human use in the United States. It's about 1,300 drugs or so for about 6,800 indications. There was then a panel set up about 2 years ago by the lupus voluntary organizations, in which suggestions for drug repositioning were solicited from various lupus key opinion leaders. Then every agent that had ever been successfully used to treat murine lupus was looked at, and finally a crowdsourcing approach was undertaken, in which a LinkedIn site was set up, and suggestions were solicited from the lupus community as well as the community of interest scientists, patients, everybody communicated. And if you're interested, you could look at this LinkedIn site. There's still a very active discussion going on and suggestions coming in every day. The initial group of compounds and therapies were reviewed. I'll show you a little bit about that for mechanistic effects, drug properties, safety profiles, and then a prioritized list was established, was vetted by a committee of experts, and then a list of high priority candidates was established. Oops. Now, in the process of this, I have to say that when you start with the social media and crowdsourcing, you have no control over who actually contributes ideas. And we had lots and lots of contributions, initially from small biotech companies, probably looking for money, but then also from larger pharma looking for support for drugs that might be useful for lupus. So we again began to expand this initial concept of repositioning approved drugs into repositioning drugs during development with either biotech or pharma, and we reviewed a lot of those drug candidates as well. So here's the process, pretty straightforward. We had community suggestions, we had an expert meeting, we searched all approved drugs, we looked for drugs that are active in, or compounds that are active in lupus, and then we looked for interesting drugs in development for other indications. And then from all of this, we came up with 157 drugs or treatments that seemed to have the likelihood of a possibility of success in lupus. These underwent a systematic review And then we had the problem of what do you do with all this information? We had more than, I think, 6,000 or 7,000 peer-reviewed papers that were reviewed, along with patents and a variety of other information. So we then decided to try to rank these drugs, and I'll tell you a little bit about how we ranked it. And so we could rank the drugs by a scoring system. Then we went to the vetting committee, and then we moved to trials. So we created this thing called the Combined Lupus Treatment Score. There is basically, at the moment, other than this, no non-biased way to relate or rank drugs for their likelihood of success in lupus. And this was done in an iterative process with a number of experts who have some knowledge about how to go about making these scores. And we created scores and then tested standard of care drugs, and then used those scoring to tweak the scoring system. And finally, we came up with this scoring system, which allowed us to look at every aspect of a drug from its rationale all the way through its clinical performance. So you can see here we could rank drugs by rationale on a 3-point scale. Again, looking at the literature, the goal here was that if everybody in the room was willing to look in the literature, they would probably come up with the same score based upon having a 0 with no role, a 1 or a 2, depending upon how strongly the role was documented in the literature. We looked in lupus mice, and again, if no benefit, you basically lost a point. If it wasn't done or was equivocal, you got 0, and a benefit got you a point. And this was based upon the fact that our colleagues in pharma counseled us that lupus mice don't always predict success of lupus therapies, so you only could get a point on a lupus mouse. Lupus cells in vitro, does the drug do something to correct an abnormality that has been documented in lupus cells? Lupus abnormalities, has that pathway been shown in vivo to have something to do with lupus? Autoimmunity, clinical experience, other diseases, does it work in a related disease? Lupus clinical experience, most of this came from case reports, anecdotes, what have you. And then we had a lot of things rating properties having to do with route and frequency of administration, characteristics, drug interactions, what have you. Whether or not the drug induces lupus, there are a lot of them that do. Metabolism, you could lose some points having to do with your metabolic pathway, renal excretion, what have you. And then adverse events, we went through all of the adverse events and developed a way to score a drug for how bad the adverse events were. So when you're all said and done, if you were a terrific drug, you could get an 11. If you were a terrible drug, you got a -16, and most drugs scored somewhere in between. So here's the testing of drugs that are available. If anybody's here from GSK, I apologize. But belimumab, this is a drug that's been approved for lupus. And you can see, it has a pretty good rationale, has very good data in lupus mice, a lot of characteristics here. It's a pretty safe drug. It winds up with a score of 5 on a scoring system that goes from 11 to -16. You can look at hydroxychloroquine, also scores a 5 on for a somewhat different route. Rituximab gives you a 3. So basically, we were looking for drugs that did better than this, something that gives you a bit better likelihood of success. So when all was said and done, what was the highest scoring biologic? We rated all drugs, biologics, small molecules, complementary molecules, drugs in development. The one that scored highest was ustekinumab, sold as Stelara. Here is ustekinumab, and it scores a 10. So substantially better than the drugs that I showed you that are standard of care drugs. And this blocks the common subunit of IL-12 and IL-23, and by doing that blocks the generation of Th1 and Th17 cells. It works quite effectively in lupus mice. Lupus cells, you can correct these abnormalities. There's a tremendously large experience with psoriasis. There are case reports in cutaneous lupus and of Stelara. It's a subcutaneous human antibody that works, it's given 4 times a year, doesn't induce lupus, has no problems with this, and its adverse events are minimal. So this drug scored, this biologic scored very highly on our list. Others, I should say, also scored quite well. But where are we with this? Well, based on this information, we went to visit our friends in Janssen, who were skeptical, but after a long period of negotiation, there's a proof of concept trial plan with first patient in October of 2015. So we're very excited about that because we think that this external drug repositioning has caused a large company to rethink whether or not a drug that they were successfully marketing might also be used in lupus. The highest scoring complementary and alternative therapy was krill oil. Now, what is krill oil? You've probably seen it on the internet. You've seen it in the health food stores, but I'm here to tell you a little bit about krill oil. This is actually a krill. A krill is a small shrimpy-like thing that lives in Antarctica, and it is the bottom of the food chain. It eats algae. Algae are highly enriched in omega-3. This thing lives at about ocean water at about 32 degrees Fahrenheit. So basically, omega-3 fats are very important constituents of all its of all its membrane phospholipids, basically in order to keep the thing from freezing. So this little organism is loaded with omega-3s. And the most important thing is, since it's the bottom of the food chain, it has no mercury in it. So you can make omega-3 oil out of this, which is both highly enriched in phospholipid-containing omega-3s and has no mercury in it. And we know that in lupus patients, omega-3 deficiency is quite common. In fact, we know that everybody in North America is deficient in omega-3s because of our diet. So why do omega-3s work? And I'll take you through this just very quickly, but there are a number of mechanisms of action that can actually be measured. It's an alternative substrate that leads to the production of ineffective eicosanoids, including prostaglandins, thromboxanes. It's a precursor for the production of anti-inflammatory lipid mediators, including resolvins, protectins, endocannabinoids, all of which have potent anti-inflammatory effects. Omega-3s also function as agonists for a number of receptors, GPR120, another of these G protein-related receptors. This one actually functions to shut down the inflammasome and block the stimulation of NF-kappa B. It is also an agonist for PPAR gamma and actually for SIRT1, as well as NRF2, which is a critical pathway that stimulates protection from oxidative stress. It also alters membrane function by basically disrupting lipid rafts, and as a result inhibits signaling through the T cell and the B cell receptor. What happens in lupus mice? Well, you can treat NZB mice quite effectively with this, or BXSB mice, and if you feed these mice omega-3 rich diet, they actually can't get lupus. There are a number of other things I don't go into. There's a clinical experience in RA. There is some clinical experience in lupus, and this drug is basically safe. It's generally regarded as safe as an over-the-counter supplement or medical food. So what's going on in krill oil? Well, MOU has been signed with a company in Norway that makes this stuff, and this is another clinical trial that will begin in the third quarter of this year. So we're also quite excited about that. Small molecules. Well, here's a very interesting molecule which we're quite excited about because this came partially from the work of an investigator-funded by the ALR looking at the role of a particular kinase, ROC kinase 2, and there's an inhibitor of this available. ROC kinase is upstream of IRF4 and is critically involved in driving the maturation of Th17 and T follicular helper cells. So, a lot of words on here. This drug is a selective inhibitor of ROC2. It basically inhibits the production of Th17 and T follicular helper cells, and also enhances the production of T regulatory cells. There's a lot of information about blocking in lupus mice. It corrects abnormalities in lupus cells in vitro. There are many abnormalities that could be targeted by this. We don't know much about its toxicity because the phase 1 studies are still going on. But in terms of a modified score, it scores very highly. Where are we? There's again an MOU has been signed with the company that makes this called Cadman in New York. And again, there's an anticipated beginning of this trial this year, which we're very, very excited about as well. So, literature mining has been helpful. It's identified some compounds, but fundamentally, the literature is not growing fast enough to keep mining. So we want to widen out the search for new drugs. And one of the ways that we've begun, which I'd like to share with you, is by looking at gene expression profiles and seeing if we can match them with drug action profiles and begin to develop new thinking about lupus therapeutics. And I'd like to share a little bit with of that with you now. So what's the concept here? Well, the concept is identified here. And that is that we're trying to find new drugs, and that could happen in a number of ways. So you have a drug with a known action, and it works in a disease. But what happens if you discover a new action of that drug? It might work in a different disease. So there are many examples of this. I mean, easy one would be, for example, tofacitinib, a JAK inhibitor that was just approved, that started life as a JAK3 inhibitor. And it went into RA because it was supposed to be inhibiting cytokines driven by the common gamma chain, so IL-2, 4, 7, 9, 15, and 21. But in fact, in development, it became a pan-JAK inhibitor, an inhibitor of JAK1, which brings into thinking the idea that it could inhibit also the interferon pathway, the IL-6 pathway, and perhaps might be interesting in a disease like lupus where these pathways are quite active. The other way that goes on is, for example, you have a known target, drug is treating with a known target, known disease, you learn something more about the disease, and all of a sudden a drug which is available might work in that disease. A simple example is, for example, recent work has shown that cancer cells change their metabolism. So they go from using mitochondrial sources for energy to glycolysis. And if you block glycolysis in a number of cancer cells, they basically die. So a drug is on the shelf. Metformin can actually inhibit that pathway. So if you look in clinicaltrials.gov, you'll see numerous trials going on in a variety of malignancies using metformin because something new was learned about a disease and an available drug was there. So the idea is to learn about the compounds, to learn about the disease, to begin to make these kind of interactive maps, to understand the interactions, and then to develop new drugs. To do it a little bit more specifically, here are the drugs do things. Here are the patients have abnormalities. Getting an integrated network can give you a new drug to reposition, and what may be even more interesting is to begin to actually develop rational drug combinations, understanding what the various drugs and pathways are. So here's what we've done. The easiest way to start is to start with gene expression profiles, although one could do this in a variety of other ways, including some of the GWAS studies, the epigenetics, but the one that's the richest are the gene expression studies, and that's where we began. So the approach was very simple, and that was to identify all the gene expression data sets that are available, and it's a lot because most journals now insist that gene expression data be put onto a public website, either GEO in the United States or ArrayExpress in Europe. And from this, you can get the raw data. And from the raw data, you can curate it, because these weren't always done perfectly. And I have to say that as a journal editor, I'm embarrassed to tell you that the method of the microarray and its analysis is never reviewed in peer-reviewed journals. So when you get the raw data, sometimes it is quite amazing. So curation is absolutely essential in order to normalize and to get rid of the variation. And then you have a number of subsets. You can undergo meta-analysis looking for consistent and significant changes using a variety of statistical approaches. You can validate it with a different data set, and then you can cross-reference it with the various drug databases, and I'll show you one of these as we go forward. So here's the data sets, and just by going to the literature, this is actually interesting. This is the ultimate in dry lab experiments, but looking, you can find that just immediately there are more than 1,000 lupus gene expression profiles available on these various websites, and probably an equal number of normals. And they come from different cell types. They come from peripheral blood. They come from tissue. They come from kids. They come from all kinds of different things. So there's a rich, rich database that one can analyze, and there are more that we will have. We expect that we'll get probably in the range of 2,000 to 3,000 of these to fully analyze when we're done. So from that, what you can do in your meta-analysis is basically begin to create a lupus signature. It's just the top 40 genes from lupus patients compared to normals, and you can see a lot of things are up and the green is down. So it's easy to make a signature of what's wrong with regard to gene expression in lupus. And you can cluster these in various ways. This is just a way to cluster these genes that are abnormal in lupus based upon their behavior in the arrays. But then what you can begin to see is that some of these clusters, and you don't have to worry about the names, the program just names them by colors, some of these correlate with disease activity in lupus very strongly. Some of them correlate with anti-DNA, some of them correlate with complement levels, but it's easy to find a fairly large number of genes that correlate in this situation with disease activity by SLE in lupus subsets. And you can begin to put these together in pathway maps. One of these is the interferon pathway. One of these is cellular proliferation pathway. There's an apoptosis pathway. And you can begin to get the idea that you can understand pathways that were heretofore not well understood by putting together the analysis of these various expression profiles into pathways. And then the question is, okay, you have some disease genes. Is there a way to get drug genes? Because that's what you want to find, is the interaction between the disease signature and the drug signature, and understand new potential drugs that could be useful in a disease. Well, there are many ways to do this. One is to look at this, the Library of Integrated Cellular Signatures. This was a project funded by the NIH, housed in the Broad Institute, and their goal is to understand the molecular action of a variety of different drugs and what they call perturbagens, alterations in gene expression. So the idea, and this is not Goodman and Gilman, the idea here is to use basically brute force. Take a bunch of cell lines, dump a bunch of drugs on, and do gene expression. And then you will understand all of the actions of the drug. So a PI3 kinase inhibitor may inhibit a very specific enzyme, but as a result, there are downstream changes in a variety of gene expressions, and you can map that out, and you can begin to tag each drug with a set of gene actions. So what's in links, and you can get this on the internet, It's not actually easy to get it on the internet, but you can. Basically, what they've done is taken almost now 15,000 compounds. It includes FDA-approved drugs, bioactive tool compounds, a variety of screening hits from companies, and they've put them onto cells, and they've measured gene expression. In addition, they've taken now nearly 5,000 genes of interest to them, and they've either knocked them down or overexpressed them, and again, looked at the effect on gene expression. And they've done this in a variety of different cell types. Since this began as a cancer project, many of these are tumor cells, but now there are a variety of other cell lines that this have been done. So what you get at the end of the day is an excess now of 1.5 million gene expression profiles, all related to either drug action or to up or down regulation of a specific gene. So you can take your little lupus signature here. There's a volcano plot, these are the genes, and you can then match it to the drug action databases through links or through other ones. And what happens is you get this gigantic list of drugs that match your disease profile. So here's lupus identified pathways, and this goes on and on because they rate everything by a so-called connectivity score. How closely does it relate to the action of that particular drug? And you can go through this entire thing. And what we've done, I've just highlighted you some because I want to show you that this actually can work. So I've picked a few of these out. And what we decided was, here's a drug, mycophenolate. That's a drug that's used in lupus. It has a pretty high connectivity score to the genes that we were able to identify as the lupus signature. It has a high connectivity score, but we wanted stuff that's better than mycophenolate. So we anchored that. And then I'm just giving you some drugs here. You know, there are some steroids up here. There's actually, where is it? There's, I have to look at it here, down there somewhere. Here's quinacrine, a drug which is actually very interesting to a number of rheumatologists. Here's triptolide from a Chinese root that we showed is very effective in lupus, and a whole bunch of other drug candidates in between. When we see this list, we begin to say this works because we know some of these work. Here's a drug, Fasenmyer. Fasenmyer is the parent compound of the ROCK inhibitor that I mentioned that we are actually moving into a clinical trial. So this approach can really work. Well, what about the gene approach? Well, that can really work too, because again, here's the same kind of thing, zillions and zillions of things, pages of this stuff goes on, and I've just picked out a few. This is what happens when you look at the gene signatures from overexpression. So you've overexpressed this pathway, and here's its similarity to what we found with a lupus signature, which says basically that these pathways are likely to be very similar to what's driving lupus. And we can see, you know, BCL10, we know that's downstream of the BCR. LIN, we know that's involved in the BCR complex. CD40, lots of these are working. And then we can look at the ones that are opposite, because these are pathways that might be candidates specifically for the lupus signature. And you can see some interesting ones here, BCL6 that drives germinal center reactions, TRAF2, a variety of things. So our view is that this is going to work, and if I ever come back, or somebody else working on this comes back, I think we will be able to tell you about clinical trials based on this kind of big data analysis of both gene expression from lupus patients as well as and cross-reference to the drug action. So it's an unbiased meta-analysis of human genes begun to identify novel targets. New and unidentified treatments from lupus patients will emerge, we believe. When one begins to further annotate this with GWAS data, epigenetic and proteomic data, we think there'll be a lot of novel pathways identified and drugs that will be interesting, and cross-reference to the known drugs and pharma pipeline databases should provide, in our view, the next generation of new therapies for lupus. So the goal of all of this data mining is to find the gold, and we're very encouraged so far that we may well do that. So thank you very much for your attention.